Hello, everyone. Yeah, let me just confirm that people online can hear us. Yeah, if you can put your hands on just to confirm that. Okay, fantastic. Okay. So thank you very much. Yeah, it is. As you know, is one of the big focus of CERI to create this really transdisciplinary seminar series. And this week we are very lucky because first is our second large seminar of this week. Yeah, on Wednesday we had Professor Wolfgang Preiser talking about how to discover new viruses and was extremely well attended. And then today we have Ewan Harrison from the Sanger, and it's fantastic to see again a completely full room. So we are very excited about all the excitement about these open seminars in this building. Yeah. So today we also have another important visitor, Professor Jeffrey. So he's our DVC of Research of Northwest University. So if you want some full professorship positions, you can talk with him after the meeting. One of his objectives is to also make NWU one of the top universities on research in the country. Is that true, Professor Jeffrey? Of course, yeah. And I also believe that you're going to be opening a medical school sooner, or is in discussion, yeah? 2026, yeah. And so it is always very exciting to see a lot of important visitors come. But today the talk is about Ewan Harrison, which is from the Wellcome Sanger Institute. You may be aware that we start a big strategic alliance with the Wellcome Sanger Institute. Yeah, I was appointed as deputy director of the Genomics Surveillance Unit with one of the big objectives, yeah, to bring new technologies and working together to really open the way that genomics is done in Africa as it has never been able to do before. Yeah. So the Wellcome Sanger Institute is just a little bit south of Cambridge. Yeah, it's the biggest genomics facility in the world. Yeah, and Ewan Harrison is the head of the microbiome and respiratory infection virus at the Parasite and Microbe program. He's also you're also a professor, yeah, at Cambridge. Or okay. So he's assistant professor also at Cambridge University. So a lot of the positions between the Wellcome Sanger Institute are co-shared with Cambridge that you get dual affiliation. Yeah. And he was very kind because he's coming to South Africa to spend a week giving a workshop on antimicrobial resistance with the Wellcome Connecting Science in Johannesburg at the NICD. But he decided that he landed today in South Africa, and he opened the day to be with us, both meeting with us and also to give our seminar. So it's quite brave, come straight from the airport to be here. And then he's going to go back to Johannesburg at 8 p.m. But we don't want to just torture him. So after here, we are taking him to a picnic in Spear, so he can at least see a little bit more of the things. Yeah. So today, what he's going to be talking, I know a little bit what he's going to be talking is really exciting program that the Wellcome Sanger Institute developed with the GSU, which are also participating, yeah, on how you could go from one swab and get multiple viruses as cheap as doing other methods. I hope I didn't steal your thunder, but we are very excited to be here. And he was impressed with you. He said that you are even more popular here than back home, at least from the seminar. So thank you, Ewan. Yeah. What do you want me to do? It's not an everybody's way. There we go.
Brilliant, thanks. Can you hear me at the back? Okay. Yeah. Yes. To the microphone. Okay. Yeah. Brilliant, thanks. So, well, it's fantastic to be here today to talk to you because the work that I'm talking about really is building on, I think globally the success of pathogen genomics during the COVID pandemic was a lot of the people who work here within this institute were we're part of that global success. And I think that the way that pathogen genomics has spread across the world since the coronavirus pandemic just shows us really, still how far we've got to go, but also what's possible if we put our minds to it. And the work that I'm going to be talking about today really is building on that success and saying, okay, we built COVID genomic surveillance capability around the world. Let's build on that now. Let's start sequencing all respiratory viruses, because obviously they're a major risk to human health. But by doing that, we keep the sequences turning. We build further development and training of people around the world for pathogen genomics. So I promise to this audience, I don't need to really go on about too much about how important that genomics was to the response globally to COVID-19. Of course, without the original genome sequence being made publicly available in January 2020, we wouldn't have had vaccines, we wouldn't have had diagnostic tests, and of course we wouldn't have cheap sequencing as well. And I think that's quite a message that's often forgotten when we talk about the importance of genomics. We kind of think about the response later on, how we used it to track variants around the world, how we used it to understand vaccine escape, etc. But actually from day one, genomics was right at the front of our response to COVID. And if we didn't have that capability, we would have obviously been in a much worse position. And so I think that's a really important foundation, and we'll be thinking about how we use genomics in the future. So what would have been even better though? We did quite well. What would have been even better? I think arguably it would have been better if we'd spotted SARS-CoV-2 before it spread epidemically. I think that's ultimately the point that we need to get to. Earlier understanding, if we'd had genomic capability around the world, we would have probably understood more about the transmission and how far it had actually spread around the world before many governments acted. So I think these are all things that we obviously want to change for the future. And of course, what things do we need to know more about? I think you were saying that we had a speaker earlier in the week about discovering new viruses, about host jumps. We need to know more about what happens when viruses jump into new hosts. We need to understand more about the evolutionary biology. Coronaviruses have jumped into human populations on multiple times over the last 20 years. We obviously need to know more about coronavirus biology, full stop. And I think obviously a key thing is that we know now that we need global capability sequencing, and that needs to be equitable. It needs to be around the world. And the bottom line, that means it needs to be cheap so that everybody can do it, and we can do lots of it so that we can hopefully answer all of these aims. Really, we can spot viruses earlier, we can understand more about them, and by doing that we need to do it at scale. So there's obviously an ongoing need for pathogen genomic surveillance. These are just headlines that I've taken from the BBC News in the UK over the last few year or so. There's been, there's obviously major pandemic threats from avian influenza or other influenza viruses, but there's continual introductions of new viruses into human populations, and we obviously need to keep better track of that. So in the UK, during the COVID pandemic, we had a consortium called COG UK, which was academics, public health scientists, and large genome facilities where I worked, all working together to sequence. And we were quite lucky that in April 2020, or even before in March 2020, the government gave us money to get going with sequencing and basically left us to get on with it, which was a great opportunity for academics, public health scientists to really just work together towards one common solution. And I know similar things happened here in South Africa, and latterly in many other countries around the world. At Sanger, where I work, we built a kind of a machine for sequencing COVID genomes. It took a while to get this built, but ultimately we could take 500,000 samples a week that came from these mass testing laboratories that were testing samples all over. So if you in the UK, if you had symptoms, you did a swab, you put it in the post. It went to one of these huge testing laboratories, and we could take 500,000 samples a week. We had, I've seen your new lovely storage downstairs. We had a slightly more quickly put together system where we had shipping containers in a car park that could take 6 million samples. But this allowed us eventually to build a system that could get through 67,000 COVID genomes in a week. And that was our kind of peak. And we had robotic cherry picking and a kind of an end-to-end tracking system. This took a time to build. but it was an incredibly effective system for COVID genomics. And really, at the end of this, we thought, well, if we can do this for COVID, really, can't we do this for all respiratory viruses? Can we use them, the mechanisms, the machinery that we've built for sequencing COVID, can we use it for other respiratory viruses? So obviously COVID is one of many, and the work that we're doing now is really to start sequencing, is to get assays that work for all kind of majorly clinically important respiratory viruses, and this is the list that we're targeting. And obviously, the more that we can learn about coronaviruses, about parainfluenza in general, is also good for basic research and public health, but also may provide us further insights that will make us better prepared for future pandemics. So in the UK at the moment, this is how kind of the state of surveillance for all respiratory viruses. It's basically diagnostic based testing. Except for COVID and flu sequencing, we just have testing for a range of viruses, and that data goes centrally into our public health agency, and we can monitor epidemic curves. And you can see here, you can see the curves of flu, RSV, and COVID. But genomics for other respiratory viruses, we don't currently have. Yes, that's this year. Yeah, up until just after Christmas. Yeah. So we wrote a proposal at Sanger to build on our work, and we were funded by the Institute to build a new kind of program, which we call the Respiratory Virus and Microbiome Initiative. So this is a kind of a bridging activity. We have a core research component, which is the part that I lead within the Parasites and Microbes program, which is to basically do the research and development, use the data that we generate for biology, but then ultimately working with the Genomics Surveillance Unit, the idea is to turn this into products. So when we develop a bioinformatics pipeline, it goes into GitHub, it has instructions, it makes it really easy for other people to use. So everything that we're building is open source and shareable by others, including the protocols and methodology. We've got two major aims. One is to generate cheap sequencing methods for all respiratory viruses that could be used during an epidemic or a pandemic, but also then we're going to study using this data, we'll study the respiratory viruses, but also their interaction with the microbiome, and I'll talk a little bit about that. Most of my talk today is about the work we've been doing on respiratory virus sequencing. So what are our principles? Well, I think for this to be effective, we've set ourselves an aim of getting it to $20 a sample or less. It's not there at the moment, but that's our aim, because we think that that's probably about the price point where it starts to be usable globally per sample. It needs to be robust. It doesn't need to rely on reagents to have a single source. I think your experience here in South Africa particularly is a really important example of what can happen if there's a pandemic and there's limited access to resources for political reasons or for other reasons. That can cause major problems to supply chains and the ability for genomics to take place. So we're trying to think about methods that aren't reliant on a single product or a single source. We're also thinking about reduction in cold chain, thinking about sequencing being done in more remote areas. It should be adaptable and customizable, and we're also thinking that, again, everything should be open access. And finally, at the moment, we're aiming to make it sequencing platform agnostic, because different sites have different sequencing technology. At the moment, we're kind of focusing on Illumina sequencing, but we're also going to start working on nanopore sequencing as well. So COVID sequencing, for probably most of you in the room, I don't need to talk about, is done in most cases by amplicon sequencing. This is great, it's cheap, it's very effective. One problem is when you get mutations in the virus, you need to change primers. That can take time to change. And of course, you're playing a kind of constant catch-up game. The approach that we're taking is more kind of metagenomics. So you can go two ways with metagenomics. You can do true shotgun metagenomics, where you take a sample, you take all the nucleic acid or DNA in that sample, and you just sequence the whole lot. And of course, the pathogen of interest may be at quite a low level of abundance compared to the human DNA or bacterial DNA that's in that sample. So the approach that we're taking is using what's called bait capture metagenomics. So we have short strands of baits that target across the genome. So if you imagine you've got a genome of 30,000, the COVID genome, you have baits that cover the whole genome. But the advantage of this is that you can use multiple baits in the same assay. So you don't just have to target just COVID, you can have a pool of baits that target a range of respiratory viruses, all within the same assay. And then what you do is you use those baits to capture the proportion of your sequencing library that contains the pathogen of interest. You then amplify that up, and then you get an enriched sequence data that contains your pathogen. You get some background still, like the method Genomics, but you get a highly enriched proportion of the data. So we think that this is the quickest route, and there are others. I know I've been talking to people today. There's others in many places working on a similar thing. This is the quickest route to rapidly expanding routine sequencing of respiratory viruses. It's been used successfully by many of the laboratories for other targets, but in many cases, cost is an issue. So that's something that we're aiming to work on. The baits themselves are quite expensive. And it also, we think it requires a systematic evaluation in the sense of determining sensitivity and specificity and benchmarking that against existing amplicon-based methods. So that's what we've been doing. So we tried 3 different methods. So Illumina sell a method for this off the shelf is about $100 in the UK, but in many other parts of the world, it's way more expensive than that, up to $200 per sample. We've taken a method that was developed in Oxford by David Bonsall and Christoph Fraser. They actually used it for COVID sequencing during the pandemic, but it was originally developed for sequencing of HIV and hepatitis. And then we cobbled together a method with various pipelines that we had running within our institute. The reason we did that is because it was the quickest way to automate it. So most of this was the library prep and everything is done with robots, so there's minimal pipetting. And for us to scale up and to demonstrate this was the quickest route. So we wanted to see, are there any clear advantages to any 3 of these methods? What are the strengths and weaknesses? And basically, does the method that we've got in-house already that we can get going, is there any obvious reason why we shouldn't use this method? So this work was carried out by our kind of R&D team in our sequencing part of the institute. And what we did is we took these 3 different methods, and we took a batch of 32 samples. So the first thing that to note is that we are using probes that target a broad range of respiratory viruses. So for the 2 methods, the one that was developed in Oxford and our method, this is the range we were using twist probes. So these are commercially available probes that target this range of respiratory viruses. So you're targeting influenza, adenoviruses, coronaviruses, the kind of a broader family, rubella, enteroviruses, so rhinoviruses, metapneumovirus, RSV, but also parainfluenza, measles and mumps are also included in the panel at the moment. And in theory, you can add into these panels and customize them. So we split a plate. The first thing we just did is we split a plate of sequencing on an Illumina run between the 3 different methods. This is the details of the different methods. We plexed for the bait capture, so you pool the samples together when you do the capture of the library. So the Illumina one, they recommended doing it 3 samples in one. For the other 2, we used 8 samples plexed together. And we used a range of samples. So this is how the plate was split, 3 different methods: Illumina, Amphius, and our in-house method. On the left here, we used a range of twist synthetic RNA controls, but then we also included a selection of real COVID samples, clinical specimens that were both positive for SARS-CoV-2, but also negative from people who had symptoms but didn't test positive for SARS-CoV-2. And then we sequenced this plate. We actually sequenced it twice to look at reproducibility. This is just to show you that this is the raw number of reads. But the one thing that you can note straight away is that in the clinical samples right away, we're generating a lot more read data than we are in the synthetic control samples, which again we thought was positive because we're capturing from the real clinical specimens. We'd be more worried if we weren't seeing effective sequencing. Yeah, so it's the one at the top. This is then just looking at the reproducibility. So this is then the number of reads from each of the samples that mapped to viruses in viral RefSec. Again, you can see in red consistently the clinical specimens were working really well across all 3 methods. And then this is using InStrain to map the proportion of reads that matched to a range of different species here. So across the bottom, It starts with SARS-CoV-2, goes into human coronaviruses, human parainfluenza 1, and then you've got two sets of flu segments, H1N1, H3N2, the 8 segments. And consistently across all three of the methods, there was no clear, they all worked well. There was no clear, weak method. So we concluded that basically, we were quite happy to take all three methods into a kind of a larger scale pilot to see how well they performed. This just shows you, this is genome coverage. So again, across all three of them, this is the flu, the 8 segments of flu. At the top, the real clinical specimens from SARS-CoV-2. Again, it was working for all the viruses, including samples where we mixed together. This one here is a sample with SARS-CoV-2, human parainfluenza 1, and flu in the same sample. And we're generating, from 30,000 copies of each, we're generating full-length genomes for all three viruses. And it was broadly similar across all three of the methods. So if we then dug in a little bit further, because we had a titration of flu in these samples. And again, so you can see at the top here, the three different methods. So at the top was our in-house one, Amphius is the next one down, and then Illumina. And then you've got 1 million copies, 100,000 copies, 10,000 copies, 1,000 copies, and 100 copies. And this is the fraction of the genome that was sequenced with 10-fold coverage. And you can see that we're down at 100 copies. We're detecting with all three methods. There's some variation there. Once you get to 1,000 copies, you're nearly getting to get pretty good coverage of genomes. When you reach 10,000 copies, you're getting a full-length genome using all three methods. So I'll come on to that later. So this, we've got some kind of more detailed. We actually looked in the kind of in clinical specimens. We matched up this viral load to CT. So a CT of 10,000 is, 10,000 copies is a CT of about 25. So it's around where you're seeing with with amplicon sequencing is a little bit higher, and I'll show you. We did that, and reproducibly we can show that it works well. This is then comparing the coverage in. So now you're looking across. This is these are the amplicons in a SARS-CoV-2 Arctic genome, and then you're looking at various clinical specimens here. The blue line is the coverage you get with an amplicon sequencing, and then the lines below are the three different methods. So the yellow one is the in-house method, the Illumina is the kind of green one, and the red is the Amphius one. And we're looking across here, of course, these were old samples, I should note. So we were talking a bit earlier about, what happens with samples in the freezer. Some of these have been in the freezer for 2 years. So we were still getting reasonable coverage genomes using this method from quite old samples. Of course, you can see, and we'll see this later, that the amplicon does give slightly higher. But of course, you've got an assay here that can target multiple respiratory viruses within one sample. This just shows this a bit in a similar thing, comparing the three different methods. So you can see slightly, in this case, the Amphius and Illumina was slightly higher coverage. So this is then comparing, this is actually then pangolin lineage calls on these samples. And what we found was in the older samples, the bait capture didn't work as well. But actually in the newer samples, so Omicron samples, there's two marked up there in blue. Actually, the bait capture sequencing gave a higher resolution pangolin lineage call because of amplicon dropout due to the primers. So even though we're getting lower coverage, that coverage is actually more even across the genome. And I'll show you later some data that really shows that quite clearly. So we then went on to kind of into phase 2. So we now scaled up to 96 samples for each of the methods and really did a head-to-head to see whether there was any difference between the three methods at a larger number of samples. And also, again, to look at the reproducibility for these sequencing assays. But we were also thinking about, again, about sensitivity and specificity of the sequencing as well. So this time we repeated using the synthetic control samples, We also included a range of COVID negative samples. So these ones here, these all came from people who had access to antivirals. So they were clinically susceptible people. So they had some underlying problem, most of them immunological problems. So they could access testing in the UK. We got their samples. So they were symptomatic for something, but they didn't have SARS-CoV-2. They tested negative. So we put those in just to see how well it would work on unknown samples. We also included a range of different controls. Again, we included some extracted virus from nibs there. You can see, so this was RSV-B and H1N1 as well. We also took we included a Zepometrics panel, which includes basically a whole range of respiratory viruses all within one control, just to see how well it would perform across multiple respiratory viruses in the same sample, thinking about complex patient samples that may have more than one respiratory virus. So this is just some things that we noticed. So this is kind of really getting into the technical details now of the sequencing is the library yield. So one of the things that we noticed was that we saw quite differences in the in the library yield when we were generating the libraries. So you can see here on the left, the Illumina was generating much higher concentration libraries, broadly similar to our in-house method, whereas the Amphius one was producing much smaller yields of library. But actually, when you go into the sequencing, we found this doesn't really make any difference because of the bait capture. So when we look at post-capture yields, so one thing I should note that we did here in this experiment that I didn't mention is that I mentioned before we were pooling 3 samples for the Illumina. That's what they recommended for us to do because that's what they said in their protocol. But we decided, because the baits are very expensive, we would up it to 8, which was in parallel with the other 2 methods. So again, we were testing whether or not we could start to increase the number of samples in a pool to reduce the cost of the sequencing. So one of the things we did notice is that we had a major difference in the insert size in the libraries. So if you look at the Illumina here, you can see the average insert size was about 300 bases. Amphius was about the same, but our in-house method actually had a much smaller insert size in the library, which we thought was going to be a problem because you get more read into the adapters. But actually, when we look at the data, it's not a problem. And actually, the smaller, shorter reads may perform better. So you can see this quite clearly here. So this is the proportion of reads from each different sequencing run, with a cutoff of the minimum read length, so either 100 base pairs or 70 base pairs, and basically the red are low quality reads that were getting excluded from because they were below the threshold. And you can see in our in-house method, which is this one here, that when you use 100 base pairs, we had a lot of the data we were throwing away, so we dropped it to 70. And then it was more broad proportional. And you can see the light greens, the high quality reads that we want to take through into the analysis. And again, although the in-house method was producing slightly less than the Illumina or the Amphius, actually you'll see the data it performs as well. So this is just mapping classifications, taxonomic classifications using in-strain across the 3 different methods. So this is what you're seeing here is samples that have got SARS-CoV-2, a range of respiratory viruses. We've got human coronaviruses in here, parainfluenza, flu A and flu B. So you can see all on the same run, we're generating, this is the proportion of the reads that map to each of those. So across the 3 different methods, they were broadly similar, all sequencing the same samples reasonably well. Though when we dig into the data, so this is now looking at SARS-CoV-2. This is looking at a range of clinical samples. You can see that the ones that are marked up here in red, there is variation between the methods, but you can see that it seems to be kind of stochastic. Sometimes one method works better than the others. So another time, another one works better than the other. So it seems that there is some variation, particularly in these kind of samples where these are the ones with the higher CT. So this is CT 24, 25, which we know is at the edge of the sensitivity. There is some stochastic variation, it seems, between the methods, and presumably that's just due to the kind of the and in the library prep, there's some biases that are generating the samples. But you can see here, we're reliably sequencing COVID across all three of the methods. This is just showing you the proportion of 1 times, 5 times, and 50-fold coverage across the genome. And you can see there's really limited difference between the three of the methods. They all work pretty well. This is the same down here for human parainfluenza 1 and 2, and including samples again that were mixed with human parainfluenza, with flu, or with COVID. Again, we're still generating full-length genomes from when you've got 2 different targets in the same sample. This is now RSVB. Again, limited differences between the 3 methods. They all look pretty good. And then this is the seasonal human coronaviruses. Again, except for Amphius, which doesn't seem to perform as well, the other methods worked really well across all of the 3 human seasonal coronaviruses we included. This is flu now. So again, this is looking at the sensitivity. You can see down at 100 copies, we're detecting all 8 flu segments. This is the number of reads, the actual total number of reads that map to each of those segments there on the right. 1,000 copies, we're getting nigh on somewhere between 1,000 and 10,000 copies. We're getting full-length genomes, again, reliably across all 3 methods. This is just now clinical samples. These are real clinical samples. I mentioned that we had some of those samples from the susceptible patients. Well, actually, 4 of those had flu, and again, we were generating nigh on full-length genomes. Of course, there'll be some. This is mapping to the reference, so the differences you're seeing here are differences compared to the reference genome. But again, clinical specimens, you know, there was no prior testing done on these. We're generating full-length flu genomes from them. The lineage calls for SARS-CoV-2, again, in some cases, the Heron, the amplicon sequencing wasn't generating a call. We were getting a call with the bait capture, but in a couple of cases, the bait capture wasn't working quite as well as the amplicon, but that's what we would expect, the amplicon being slightly more sensitive. This is just showing you the mapping across the different genomes. So in summary, we found basically all 3 of the methods work pretty well. We found that increasing the pooling from 3 to 8 has a very minimal effect on the sensitivity. And actually, when you dig into this data, I haven't really gone into it to the extent. Actually, our in-house method was probably the most consistent for sequencing all the viral species we looked at. And certainly in the flu samples, it was probably the best. So we decided to take our in-house method forward to scale up. So we started to we put it into the kind of robotics now. It's gone into our kind of pipelines in our core facility. And now we're starting to train people so that we can ramp up the scale of the sequencing. And also now we're starting to work to reduce costs with a view, basically, so we're going to start increasing the plexing. So we're on 8 samples now. We're going to try and increase that more, but also we're going to dilute the baits because about 1/3 of the sequencing price is the baits. So if we can, even if we double the dilution, we're saving $10 or $15 per sample just by diluting the baits out. So the final thing that we have to do is to take it to scale, and in terms of increasing the ability to do this in a high throughput, we had to do some further experiments. So when we were doing the original sequencing runs, we ran it the reaction volume. So again, another way of making it cheaper is running a smaller reaction volume, less reagents. So we were running a 1.6 times reaction volume, which matched the 3 other methods, so they all 3 were equal. But actually, all our robotics for doing this use a smaller reaction, so a 1 times reaction. So we needed to test whether that would affect if we went down to the 1 time, so we could scale it. But also, I mentioned we saw that problem where we saw that the fragment size for our libraries was a bit smaller in this. So we thought by doing a harsher cleanup, we might be able to improve that by getting rid of those small fragments and just keeping the part of the library that we were more likely to sequence. So we tested both of these in an experiment. So again, we were also really interested to do side-by-side experiments, same plate, repeating samples basically side by side to see if we got any differences. So again, we've come back to the same pool of synthetic controls, but this time we've now got serial dilutions of RSVA, SARS-CoV-2, flu as well, and also again a bunch of clinical specimens from a range of different CT values. So this is how the kind of the process actually works. So we were generating our library prep. We were then creating our pools of eight samples were pooled together for the capture. And then 6 samples were then paired together, either 1 times or 1.6. We then did the capture and the wash, and then we did a further cleanup extra on some of the samples, an SPR cleanup, to see if we could improve that that fragment size as well. And then we sequenced them side by side on an NovaSeq SP lane. So one of the things that we found, because now we were doing the same things side by side, was that one of the post-capture, one of the pools dropped, the yield of the library dropped, and we've seen this more recently. We've had a similar issue with this, and we've now found that just by doing a couple more PCR cycles after, you can get the libraries back up. You sequence them, and they sequence really nicely. So we detected some variation that we were quite worried about, that it wasn't reliable, but actually you can just overcome that with some further PCR cycles. So again, we looked, did our change? So this is the original one. This is what we'd like to go to for the 1 time. These are the one time, so this harsher cleanup, the 1.6 with the harsher cleanup, to see whether we got any improvements. And broadly, we got roughly the same number of, if you look in there, the same order of magnitude number of reads. So going down to a smaller reaction didn't really make any difference. So that means we could automate it and begin to scale. And we also found that the cleanup process to try and clean up that didn't have really any effect either. So again, we decided not to include that because it would have added further costs to the sequencing. This just shows you the actual high quality reads compared across the 4 different conditions, and you can see they're broadly similar. So now this is now looking at reproducibility of sequencing. So now you can see here, we've got a titration of SARS-CoV-2. Again, it's similar to what we've seen for flu. Down at 100 copies of SARS-CoV-2, we're detecting it. 1,000 copies, we've nearly got full-length genomes, as using Swiss controls, and consistently there across all these. So these are arranged that start. The lowest one is a CT of 11, going up to CT of 24. Full-length genomes for all of those across, and you can see there was no effect really of any of the different methods. And up at the top there, you can see that there's samples with mixed SARS-CoV-2, human and human parainfluenza 1 and flu, and again we're generating full-length COVID genomes from those samples. In terms of lineage calls, we compared those samples to amplicon sequencing, and in this case they completely matched across the board. High quality genomes from either the BAIT or the amplicon sequencing. And importantly, this was done at the same time, same sample, same week. We took the samples head to head, and they worked really well. Although the one thing you can note here is on the high CT samples, you can see in green the amplicon sequencing is generating higher coverage, as you'd expect. But then if you look at the evenness of that coverage across the genome, you can see here in purple, that's the amplicon sequencing. You can see that there's amplicon dropout, whereas in the BAITs, it's smooth all the way across, showing the clear advantage. Although it's slightly less sensitive, you don't have to worry again about amplicon dropout. So this is then just going back into the flu. So again, reproducibly, 100 copies, we're detecting it, 1,000 copies, we're getting towards full-length genomes, 10,000 copies of full-length genomes, and again, between any of the differences we applied, there was no differences in the scale up. This is H1N1. This is looking in, again, different copy numbers between the different methods. There was no difference again. And then finally, this is showing you the reproducibility. So this is sequencing of flu in the 3 different pilots. So we included it 3 times, H1N1, pilot 1, pilot 2, pilot 3, and you can see here, really reproducible. There is some variation down at the very low at 100 copies, but once you get to 1,000 copies, it's pretty much consistent that you're, between 1,000 and 10,000 copies, you get a full-length genome. So what's really nice about this, and I think this is really important for kind of this being used in other locations, all 3 of these were done manually. They weren't done using robotics at this stage. It was still done manually. So it just shows that this can be reproducible, even if you're doing the library preps by hand, which is quite important for thinking about sequencing, around the world. And then finally, now this is looking into the seasonal coronaviruses and also RSV-A, and you can see here that 200 copies of RSV-A, we were getting nearly full-length genomes from using this method. So again, for flu, COVID, and RSV, it's working really, really well. It's just don't know why that's duplicated itself. So this is just showing this in the same way. This is just our different databases. But then finally, what we did is we did the correlation between the CT to viral copy number. This is what I was talking about. So at 10,000 copies, where we're pretty much getting a full-length genome, this was matched to a qPCR done on the same samples. You can see that we're looking at a CT of about 25, a little bit higher for RSV-A in this assay. At 10,000 copies, we know we can get a reliable genome. We also see the same in the clinical samples. So we know roughly our limit of detection is around 10,000 copies, and now we can use a CT value to decide whether to sequence samples or not. Yeah. Just not get a full genome. Yeah. So down at 100. So you'd still. Yeah. So I didn't show it. We had samples that were tested in a laboratory by qPCR for COVID, were negative for COVID under the threshold of the thing, but we detected reads across the genome. So they just didn't pass the threshold of the testing. Yeah. So this is now we've now built a taxonomic profiling pipeline. So now the data basically comes off the sequences, gets human read removal, and goes into a taxonomic profiler, which generates these outputs. And we're building a linked, customizable reference database. So the idea is that the sequencing will come off taxonomic detection of the virus. That then triggers the mapping and generation of a consensus genome using the reference genomes that have been selected from the reference work. This is work that's going on in the genomic surveillance unit. This is just showing these are real clinical specimens. So now we've taken it into production. So these were flu cases from the hospital in Cambridge. So you can see here, sequencing flu B, and then the dark blue there shows you the proportion of the genome that was completed. So that's flu B, H3N2, we're getting full-length genomes, and H1N1. And these we had no CT value for these samples. They were just patient samples that we put in that we knew were positive for these viruses, and we were reliably generating full-length genomes. We can take this out. That's using a customizable database. That was just flu, RSV, and COVID. We've got a similar one now that looks at basically the broad viral RefSeq database, and we can generate. We get these these maps that show the detection of the number of reads that align, and then the coverage. And you can see here, if you look at the top here, these are samples including seasonal human coronaviruses from patient samples, parainfluenza from human samples, and we're reliably generating full-length genomes from all of those clinical specimens. So we've taken it now from kind of the benchmarking into reliable. These samples as well, I should add, weren't treated in ideal handling. They were in a cold room at 4 degrees. We got them 4 weeks later. They then went into a -20 and then into a -80. So it shows that it's still quite robust, even with samples that have not been extracted straight into -80. There's some degree of robustness in the sequencing. So what we're going to do with it now. So as I mentioned, over the course of the pandemic, we had a lot of samples that came through the Sanger Institute. So we got rid of about 5 million of them. We've kept about 1.5 million. So we've got 1.1 million from April 2020 to April 2022. We've got 500,000 negative samples from winter 2022-23 that were from testing that people had symptoms, but didn't have a positive COVID test. So we've started to go into those, and about 30% of them are positive just straight off of sequencing, so we can get something out of them. But we also have some large studies that we're doing with the UK Health Security Agency in their CERI cohort, which is healthcare workers. And we now have a large sample of 30,000 samples, including about 4,000 samples that come from people who had respiratory viruses, respiratory symptoms within 3 days of their nasal swab. And we're going to start going into those and sequencing them to understand what's causing the infections in those individuals. And our aim is obviously to begin now to start within the populations and the samples that we've got to start studying all of these viruses, and hopefully again in the same way that you've done here, linking up with immunologists to see if this data can be useful in understanding more about vaccination strategy, etc., and evolution of these viruses. Finally, just a little pitch on the microbiome side of things. We also have a big microbiome effort. We've just finished sampling 20,000 blood donors from across England, and we're now undertaking studies that are combining the microbiome work both with human genetics to look at the interaction between the nasal microbiome and host genetics. But we're also really interested in this question about how does viral infection affect the microbiota, and does microbiota have any protective effect in the severity of infection or your likelihood of getting an infection. And again, in this cohort of healthcare workers, we've got longitudinal samples from the same people. So we're going to be doing the bait capture sequencing to look at the viruses, and then do shotgun metagenomics to go in and look at the nasal microbiome as well. And that's really this concept of how does a viral infection impact our nasal microbiome. And so I think this is a paper from an author here, which I always think is an absolutely amazing figure, which just shows the interconnectivity of the world. And I think that what we're hoping is that this work, along with others, can contribute to starting to get a sense of this for all respiratory viruses, that how they transmit around the world, how does global travel impact on the transmission and spread of viruses. So finally, I have to thank the team people who've done all the analysis you've seen today, those huge figures. Some of them are not particularly brilliant for for presentations, but I hope you get the understanding of that the data is certainly looking good and is reproducible. There's also a really huge team of people at the Sanger Institute who've been working on this, particularly within our sequencing R&D team. They're a really, really incredible group of people who've been helping. And finally, we have obviously David and Christoph for letting us use the Amphios method, our collaborators in the UK Health Security Agency, and we also have a scientific advisory board who give us advice on the direction of the RVI programme. And I'm happy to take any questions.
Thank you. So I'm just going to highlight one or two things, and then we take for questions. Yeah. So that's something, especially for our universities in South Africa. Yeah. A lot of time we talk about how to move research to innovation. This means to things that can become products. Yeah. But a lot of times we forget that the very detailed validation that you have to go through. Yeah. So and that at the moment is a big gap in South Africa. We are very good on science, or relatively good on science, but we take very few things to the market. And a lot of that is not to think about this. How do you compare methods? How do you make them reproducible? How you make them in different samples? So that's one of the things that excite me a lot of working with the Sanger and the GSU, because they're gonna take this method now, hopefully now in the next few months, nationally in the UK, and potentially sample hundreds of thousands of people as your winter thing. So before that, you have to do this very strong validation, and that's something that we have to become quite used to South Africa. And I have here DVC of Research and Innovation, a Vice Dean. Yeah, how to train our people in this very strong validation process that's done. So anyway, I'm biased because I run a lab. Our lab, the idea is to be an industrial lab that has to do that kind of things. Yeah. So just that was just my comments. Yeah. So let's open for questions. And one thing that we have from this seminar series, yeah, is that normally we grill the presenters. Yeah. So don't be shy just because he has an English accent or whatever you call that. Yeah. That with global baddies. Yeah. Especially because as we discover variants, and that's quite cool just to grill them. This analysis was quite interesting because both the Omicron BA1 and BA2, yeah, that was discovered in South Africa. And here's the red, it is who received it before. Was mostly export. 90% of the export in the world of Omicron come from North America. Who was punished for discovering Omicron? Yeah. Yeah. And then the BA2 that follow the second wave, it comes from Asia in blue. Yeah. So just to highlight that a lot of times things not what you expect. Anyway, we are proud of this paper. I hope Rudy is in the call. And that's one of the things. How can we apply that for all kind of respiratory virus? Because sometimes the ones that discover doesn't mean that's the ones that really spread. But that's not question for us or for Rudy, but for Ewan. So please, if you can just ask questions, not only about how he's going to apply, how develop method, and how can we work together. So thank you. Yeah. Mariah, yeah. Just a second, because for people. And then we're going to have questions for people online. Yeah.
Yeah, thank you. Very exciting to actually see this validation process as we're thinking of doing something similar here. I'm also very interested in how you're going to apply this in terms of surveillance, because during COVID, you're saying you did 67,000 a week. But surveillance is often difficult to fund. I'm wondering, like, what is the number of sequences that you think you would need to do to for all the, because you're doing all the pathogens at once. So yeah, what kind of magnitude do you think of in future?
Sample sets. So the CERI study that we've got, we're going to sequence so there's about 3,500 people who had symptoms over basically the winter this year. It will end in March, that had symptoms but no flu, COVID, or RSV positive test. So we're going to sequence them. We think that's a pretty good start. We've also got other samples that we can go back to in previous years, but we're looking to get we're trying to see if there's a survey that takes place in family doctors in the UK that there's about 10,000 samples that are positive per winter, and we're starting to see if we can open negotiations to get those samples, because they're a true snapshot of the viruses circulating in the community. So people basically go into their GP. If they've got respiratory symptoms, they ask them to take a swab, it gets tested. We think that they're the kind of sample sets that we can make use of. And then we do the modeling to ask the question, how big, what's the right sample size?
Yeah.
Yeah, thanks for the assay validation and setting it up. Always take time to get an assay working. My question is, when you were doing these viruses and you target all the samples, human samples, why did you not target either animal samples? Because we have spillage always come from animals to human.
So in the it's a very good question and completely right. So the major focus is on human samples, but to ask that question, we've just got a load of animal influenza viruses from our Animal Public Health Agency, and they're going on a plane, I think, this week. That we're sequencing them to make sure it works well on the more exotic influenza. But then ultimately you could expand the panel range. But at the moment, we think the beauty of this is that the baits work. So if you have a sequence that's about 80% identical, it will still work. So that means that even if there's conserved, the RNA polymerases, etc., are quite conserved. So even if you have a novel virus there, you'll still generate sequencing data that will tell you you've got a novel virus in that sample, and then you can always go back to it and do a shotgun on that sample. But yeah, I think animal viruses is we can only do so much, I think.
Thanks. That was a nice presentation. I've got two questions. One is just quick and technical. When you compared the results from the amplicon analysis, what proportion of the reads were chimeric sequences?
Can't tell you.
Okay.
I haven't looked at that. I can ask someone to look at it, but I haven't looked.
Okay, it's okay. And then the second question, I'm curious, for the study on the microbiome that you guys are about to launch, you wanna, as I understand, wanna look at the relationship between the gut microbiome and genetics. What are the broader
nasal microbiome?
Okay, what are the hypotheses sort of driving the study? And if you're interested in looking at genetics, is the UK the best place to do it? Human genetics, that is.
So reason we want to look at it is 30% of people probably in this room, I would presume it's, well, we don't know whether it's conserved in human populations. It does vary. So 30% of people in England in our study population are persistently colonized by Staphylococcus aureus, right? And that phenotype seems to be pretty consistent. And one of our hypothesis is that there is a human genetic component to that. It may not be the primary driver of that, but there may well be a human genetic component in the same way that there are certain species in the gut that are the human genetics contribute to. So our hypothesis either is to rule it out, that there isn't a human genetic component, in which case that it's something about microbial succession or immunology or the interactions of the microbiome, but we want to test whether the role of human genetics. So we've done a study that's powered to either way, we'll have an answer whether there is a role or there isn't. I think the question whether the UK is right, we did it on the back of blood donor studies where they've done the human genetics. So we made use of existing human genetics data to be able to do, the sampling itself cost £2 million. So the genetics came for free. So it might not be the ultimate thing, and we are very interested to replicate it in another human population. If we do find it, that's be the first step, would be to replicate it.
And then are you looking at environmental sources as well? Because I'm assuming for something like this to make the direct relationship, you'd need to look at air quality, maybe soil, and look at the environment around those individuals.
So we've collected information about their exposures to some degree. So animals, healthcare, we know that exposure to healthcare environments can affect Staph aureus. We've got various information about their medical and life history, their medical history. We've got access to their electronic health records, and we can track them. We are just linking to weather data, longitudinal weather data from the UK Met Office, so we can look at the environment in that scale, humidity. But in terms of the soil, no. It would obviously be it would be, and household swabbing would be really nice.
Just to highlight, I have a few questions online. I'm gonna ask to learn. They also have a project that made the CU is in Bangladesh. That's a Wellcome grant that they are measuring some microbiome on the sea or something like that. Yes, to see the difference of climate change or something like that.
Raising sea levels, you get salt.
Would be great if you chat, because that's the kind of things that interfere, no? Yes. Okay, let me take the questions online. Yeah, and then we can go more here. Yeah, question. How is the pathway from your results to public health operationalization and governmental response in the UK? This is, how long would it take for results to translate to vaccine updates?
Very good question. So we are now putting the sequencing to the point where we'll be able to do every week, regular sequencing. The turnaround time is something that we'll then work to bring down. But I think in terms of vaccine updates, obviously sequencing the earliest samples each year within the in the winter. So sequencing the earliest flu, RSV. That's would be our plan. And if you get that from the community, it would potentially be earlier than hospitals. I think, of course, That's why we need the global system. I think as much as anything, because the updates to the flu vaccines are driven predominantly what circulates in the opposite hemisphere.
Yes.
But the idea is that this data will then feed in to the kind of standard surveillance that's going on, looking for whether there is any evidence of particular, a particular subtype or clone or lineage that's growing faster, which may suggest it's got some vaccine escape. So I think the matter is routine and then at a scale where you can apply the methods that we have for COVID.
Okay, and one of the objectives with the strategic collaboration with the Sanger, it's as this method mature, which seems to be mature now, that we also starting at, we do similar method here in our winter wave. We learn from that, and that's adapted for the winter wave, which is the opposite. So we get these 2 cycles of development during the year, which potentially could help the whole world, because at the moment you develop a method and you almost wait a year for your next winter wave. So that's one of the very nice objectives. And the next question, thank you for your great presentation. What do you recommend for nucleic acid extraction from metagenomics? RNA and the DNA separately through extractions, or both RNA and DNA?
So
hello. Will this affect sensitivity of metagenomics or be the capture protocol? Thanks.
Very quick answer. If it's anything to do with bacteria, bead beating step is the agreed essential component. We've got one particular method that we're doing for our microbiome work. For the viral work, we're using the leftovers from testing platforms in hospitals, so it works well on. In most cases, they've been extracted using the Thermo Fisher Kingfisher kits that have been used. That's what we've been using, and that's the residual nucleic acids. For the bait capture. But for the microbiome samples now, we are specifically testing them head to head. We're doing a pilot experiment actually at the moment, where we're taking samples in VTM versus samples that are taken in amines transport media, extracting them 2 different methods, and then comparing the shotgun data to see whether there's any biases from the media, but also whether there's any biases in the extraction methods as well. So we'll be able to answer that shortly.
Okay, so
right now it works fine on, bog standard RNA. It's extracted for diagnostic testing.
Okay, good. So another question here. So sorry, we like grilling the speaker. It's traditional from this workshop, or this seminar. With regard to your pipeline you are developing for genomic surveillance, you mentioned bioinformatics, and I'm curious about what software you might use for your analysis. Are you using any kind non-software, or are you also developing new software, and is that become also available as open source, or you keep for yourself?
No. So it'll all be made available on GitHub, and we'll publish everything. But we're adapting. So the taxonomic profiling has been done using Kraken, but we're building a custom database at the moment. So we're going through RefSeq, and we think we need to generate some genomes to put into it, because for some of the species already, we think that there isn't a suitable reference genome in there. So we need to add one in. And then we're also using InStrain, and we've looked at some different options using InStrain for some of those figures we generated using that. And we found that particular settings are good. And again, but it's all going to be tied into one pipeline, so that which we'll publish. So it'll be. And we're also doing a whole load of work on human read removal, and we're benchmarking 3 different methods to see what's the most harsh way of removing human reads, so that when we release the metagenomic data, we don't release any human reads. So eventually there'll be a pipeline that has all the different aspects in it, and it will all be made publicly available.
Okay, sorry, I think that I have another 1 or 2 questions, and then we promise to treat you well, otherwise that's torture. It is impressive to see how the COVID sequence capabilities have been adapted to other purposes, in this case, respiratory viruses. From your experience, what insights can you share with laboratories in Africa that also have similar capabilities and should also deploy such for other purposes?
think a lot of the things we learned about doing genomics in the pandemic work for anything, and I think that maintaining one of the things that I've found is that things that we learn, the problems that we're facing now when we're trying to do it for multiple pathogens are the exact same problems we were up against, you know, and everybody was during the pandemic, you know, linking samples to patients, all these kind of things. It's those links that are really critical. And I think the link with the public health has been incredibly important for us to be able to do all this, because if you can't link the sample to the person, it becomes really hard. But I think that the technology and the approaches we've developed for COVID just needs to be lifted, and it will work pretty well. The same materials, same samples, they're all you know, perfectly usable for other viruses as well. So we've built the core, I think, is the key thing. We have the core already, we just need to build on it.
Yes. For example, here we took the COVID sick, we took the primers, we threw in the garbage, and then we replaced them with dengue, chikungunya, and worked like a bomb. So the same process. It is that people that COVID, now you can sequence that in real time. I think this week we got a whole lot of new genomes from Ethiopia of a new introduction there. So that's the kind of things that we can learn. And Lucius, before he go, talk to him, get these methods, and let's see if we can also implement that between our winter wave here. Okay, so last question, or you guys can grill him more if you want. How long does the bait capture method take from the initial sample to nucleic acid just before to sequencing? So just the process that you get a sample until you can put in the sequencer. Theoretically, how fast this could be if used for clinical diagnostic? How quick we could go from sample?
The Illumina process is about 4 days at the moment. It takes time. But if it went on to nanopore, there's a nanopore protocol that I think is much shorter. So I think that's so one of the things that we're looking at is that Illumina have got a new library prep that takes, I think, about 6 processes down into 30 minutes, just a one tube single reaction. And one of the things that we're going to do is look at testing that, because it's about the time. The thing that takes time is the bait capture. That runs basically overnight. So reducing that time, I think, is but again, it's all the things that we've got to look at.
Okay, and just because my last question, because we have the people from the biorepository here, how happy would you be with a robotic biorepository for your testing there? Very happy. So I hope the CEO of Sanger is listening.
It'll be the lab team who will be incredibly happy.
Okay. Okay. Thank you so much, Ewan. Sorry, we have our special guest with the last question.
Students have asked all the questions. I'm happy to ask a question. I don't want to compete with them because I don't have a future. They've got the future. But no, thanks. Very interesting and insightful. Just for my understanding, obviously, when you do this type of diagnostics or this tool, there is a difference to apply it for surveillance and maybe for clinical management. Can you just enlighten me? A tool like this one, will it be more suitable for clinical management or just surveillance? And I'm saying that because I think one of your rationales, you mentioned something about cost. Cost is one of the important factors that you need to consider. So taking everything into account for clinical management, do you feel that, this will be, the future for clinical management?
So I think now it's surveillance. I think at the moment, what I presented today is a surveillance tool. I think that there is use in, obviously there's a use in kind of infection control.
Yeah.
And one of the sub-studies we're going to do is we're sequencing basically all the samples from a hospital over a year to look whether there would be a utility of this for, obviously people have done it for COVID, people have done it for flu, but for the other respiratory viruses is the utility of understanding about transmission in the wards, thinking about, high risk, vulnerable individuals. I think they're the two. So general surveillance in the population, understanding biology, transmission.
Okay.
But I think in terms of the future, these methods, if they're fast enough, people can do metagenomic sequencing end to end in 6 hours, which is obviously superior to some things than culture, and as good as almost as good as a PCR diagnostic. So at the moment, I don't think it's going to replace it, but I suspect that the technology is heading in that direction. That these will be, when appropriate, but I think ultimately these are tools we want to have, right? We want to have somebody with a sequencing next to an ICU when you get a patient and you don't know what they've got. It's on that machine within 20 minutes, right? That's what we want, and I think we'll get there ultimately. But I think at the moment what I'm presenting is more of a surveillance and tool for scale, if you like.
Okay. No, thanks. Because obviously the next question is not important anymore, because I was going to ask about how you attribute, what is actually the causative pathogen if it's for clinical management.
We were talking about that just when we were having a chat earlier. We were just saying, we need it's an update. I think there's been proposals before about an update to Cox molecular postulates. You need a genomic one, and I think now we need a metagenomic one. Exactly what does it mean in those patients when you detect these, and that's why I think we need large scale studies that will build that data set over time, particularly comparing to severely ill people, moderately ill people, people at home in the community, healthy people. Will build those data sets and that understanding.
Yeah, because you think that one day it will happen, like not we. Yeah, we had many doctors here, or for example, Mariah ID specialist, that's not gonna do what they love doing. That you go to a doctor, yeah, and then they look to you and say like, "Ooh, you have a viral infection, but is here some antibiotics?" Yeah. Do you think that one day, not you, Mariah, I know that you don't do that, yeah, which antibiotics doesn't work in virus, but just to cover the bump, yeah. So you think that one day we may have that? I imagine, Jeffrey, that's going to be just a couple of years until these methods are really in the clinic, yeah, that you can detect very quick, yeah, not only the pathogen, but also to use that for very good management. So I think that they're gonna come quicker than we expect, yeah. And part of that is that kind of very clear validation that you don't get a false positive or negative, yeah. But thank you very much, Ewan, yeah, and thank you for coming, and thank you for everyone to attend here today. Thank you.
Good. That was great.
Very good.
Yeah, really good.